Hello students, good afternoon everyone, and in this lesson we are going to do the revision few important topics about the first chapter and second chapter, okay? And we are going to conduct one exam, so that is I am completely about multiple choice questions, MCQ, online MCQ questions. So I will tell the date and time about the exam, okay? But remember, so the MCQ questions will be there, multiple choice questions will be there, and the questions will be 20 questions, okay? 20 questions into 1 mark is equal to how much? 20 marks, okay? 20 marks will be the question paper. Everything will be the online, okay? Online exam, and I think you have written already. So just I will send the link. Just click on the link, and you can write the exam, and so on, okay? So while coming to the topic, so we are going to focus only on few topics, okay? Only few important topics, and these topics, the question paper will be prepared from these topics only. So those topics we are going to discuss in this lesson, okay? So by the way, good afternoon everyone, good afternoon, and welcome to the new class. So the first question will be the using scientific process skills. So what is meant by scientific process skills? Scientific process skills is nothing but, so these are the tools, okay? So these scientists are going to use the tools. These tools are nothing but utilizing the scientific processing skills to classify, compare, observe, everything. So these are nothing but the tools that is used. So processing means you are going to solve some issue, or you are identifying and solving and studying something, okay? That is known as processing. And here it includes observation. So observation means you are using the senses to learn about the object, okay? So the exam will be the, okay, Sheikh. So the exam will be the MCQ, biology MCQ, okay? So I will tell the date and time, okay? So I observe, okay, using signs, senses to learn about the object. What is meant by senses? Senses is nothing but eyes, ears, nose, tongue, and skin, okay? So these senses are used for the studying purpose, for the observation purpose. With the help of eye, you are going to observe. With the help of ear, you are going to listen. With the help of nose, you are going to smell. So it means these sense organs like eyes, ears, nose, tongue, and skin are used for the sense. Okay. So next one will be the compare. So you are identifying the characteristics between things. You are comparing between these two. It means if I give you two fruits like apple and also banana, so you can compare what is the shape, what is the color, what is the taste, okay, and which one is good to eat, and so on. It means you can compare between the two things, okay? That is known as compare. Next one will be the classify. Classify means organizing the objects or events into categories. So for example, I will give you different types of chalk pieces, okay? Red color, blue color, yellow color, and so on, and with different heights like small height, long, medium height, and also long height, okay? So you can classify the chalk pieces based upon the color and based upon the height also, okay? So you are putting in the groups: red color one group, blue color one group, yellow color one group, and shorter ones one group, longer ones one group, and medium ones one group, and so on, okay? So this is about classifying. Yes, Sheikh, the exam will be online, okay? So it means you can use the exam will be multiple choice questions, 20 marks, 20 questions, 20 marks, okay? So I will send after the completion of exam, okay, you can see your result, okay? So anyway, you can write the exams tomorrow, or I will send the link also to you, okay? So the classifying, you can classify the organs into the different various types, okay? So reading to learning, how can you read? You can read any topic, okay? You can read any topic, and you can, after reading, you can analyze, you can understand, and you can write on your own, okay? That is one of the main important things. For investigators, the scientists, and so on, reading, writing is most important, okay? So the vocabulary is most important. What is vocabulary? Means meanings. Vocabulary is nothing but meanings. So if you know proper meanings, you can learn and you can write better, okay? So before reading, what you do? Before reading, you have to think, okay? You have to think what you are going to do. So finding out, for example, if you want to find out the parts of ecosystem, you can think So vocabulary means defining each word, okay, about glossary, pronunciation, each word correctly. That is known as vocabulary. So think is nothing but finding about the parts of the ecosystem. You can find about the different parts of the ecosystem. What are living things in the ecosystem? What are non-living things in the ecosystem, and so on. So this is about before reading you can do it. Okay. So yes, from this topic exam will be, yes, from this topic only, whatever we are discussing now, from this only exam will be there. Okay. So questions will be made from these topics only that we are discussing now. From whatever we are discussing now, from these points only, question paper will be prepared. Okay, only direct questions. Okay, not complicated. Okay. So during the reading, so finding the groups of things, environment in the ecosystem, and so on. So these all of the things can be found in the ecosystem. Okay. So finding in details of the paragraph, and also writing few lines, few living things, organisms, and so on. So just now we have discussed what is non-living things, what is living. Okay, what is living? Living things are nothing but the organisms which have life. Okay, organisms which have life, that is known as living things. What is non-living? So what is non-living? Non-living means the organisms which do not have life, no life in it. For example, wind, water, rock, etc. They do not have any life, so they are non-living. Okay. So next is during the reading process, you can find out what are the things present in the environment of the ecosystem, and details of the paragraphs, and also few lines of the organisms, and so on. Few lines on the few living things about the organism that you can find. Okay. Okay. So see, Sheikh, you don't have to copy all these things. Okay, you have to copy the notes. Okay, already every day we are discussing. No, today we are doing the revision only. Okay, so every day we are discussing the topics. So that you have to write in the notes. You don't have to copy this one in the notes. Why? Because this is a revision. You don't have to copy the revision. And if you want, you can copy. That is no problem. You can copy. But so the questions will be from the topics that we have discussed. We are going to discuss important topics now. From these important topics only, questions will be prepared. Okay. So finding out the basic needs of the ecosystem, organisms living in the environment, and so on. So this is about the during. Okay. After reading, what you have to do? After reading, summarize. After reading, you can summarize what you read. Okay. For example, if you read 2 pages, okay, so 2 pages. So you can, after reading, you can summarize and you can write in half page. Okay. It means your understanding. After understanding, you can write. Okay. That is known as summarizing. Okay. And next is ask yourself about the different kinds of ecosystem and interaction and so on. Okay. So this is after reading. Now study the paragraph and also illustrations, or photograph and illustrations. For example, you can see in the ecosystem, you can see the photograph. Okay, by looking at the photograph, you can write something. Okay, how many animals are there? What are different types of animals? What are the living things in that photograph? And what are the illustrations and so on? Illustration is nothing but drawing. Okay, remember, illustration is nothing but drawing. So this can be done. So next is writing to communicate, means how you can communicate with other scientists. Okay, that is main important thing. So if you are a scientist, how do you communicate your research? Okay, that is the most important thing. Scientists to write about what they learned in their research, and also help others to understand and their work. Okay. So next is informative writing. Informative writing means you may describe your observations, inferences, conclusions, and etc. Means what you are doing, what you are observing during the experiment, that you can write in the observations. What inferences? It means what your result you get. What you can conclude, means what you can identify, what you can decide. That is nothing but so informative writing. You are writing about your experiment. Okay, that is informative writing. So next is you can tell how to do an experiment in a narrative way and so on. Okay, describing the things, giving examples, or telling the story, and etc. Everything comes under the informative writing. Okay. In expressive writing means you may write letters, you may write poems, or you may write in the form of poems, songs, etc. So this is known as expressive writing. Okay. And next is in persuasive writing. In persuasive writing means you may write letters to important issues. You may write letters about important issues in science. Okay, writing about what you have learned in the science, help others to understand everything. So this is about persuasive writing. Means writing in detail is known as persuasive writing. Okay. So next is science, safety in science. What is safety in science? Means while working in the laboratory, doing research, while you are doing or working in the laboratory, so you have to do some kind of a precautions. Some precautions is most important. So that is think ahead. Okay, thinking ahead. It means what you have to do before the experiment, before the investigation, what questions you have to find out, that you have to read. So understanding the safety protocols are shown. Okay, so if you want to do about an experiment, if you want to do some experiment, you have to find about it. You have to think about it. What you have to find out. For example, if you are writing about the photosynthesis, or if you want to do for research and photosynthesis, you have to make some questions. Okay, like what is essential for the photosynthesis, what will happen after the photosynthesis, what is the importance of the photosynthesis, all these questions. Like these questions you can make. Okay, that is about thinking ahead. Okay. Next is beware, be aware. Okay, so here you can see keeping your work area clean. If you are working on the in the laboratory, okay, doing research in that conditions, whatever the place that you do research, the place should be clean and neat. Okay, and be aware of long hairs and also sleeves. Okay, this is sleeves. It means your clothes should not interfere with the experiments. So if you have the long hair, make sure that you have to tie your hair, and if you have the long sleeves, make sure that you have to fold your sleeves and so on while working in the laboratory. Why? Because they may interfere with the chemicals and something may fall down because of your hair and some because of your leaves, because of your sleeves, something may fall down. So be aware about your belongings. Okay. Next is oops, that is inform your teacher in case of breaking something or cutting something or something damage. If there is something problem in the laboratory, so just directly inform to your teacher or inform to the lab in charge. Okay. So next is watch your eyes. Okay, while you are working in the laboratory, some kinds of acids release some kind of fumes, some kind of smell will be released. So you have to protect from the fumes of the acids and also gases which is coming from the acids. So in order to protect your eyes, you have to use the safety goggles. This is not about the goggles that you use in the fashion industry. These goggles are nothing but used for the safety. Okay, these goggles are known as safety goggles that is used for the safety in the laboratory. While working in the laboratory, you have to use these safety goggles. Okay, but not outside. Okay. So next is the yuck. It is nothing but you should never eat. No eat. Okay, do not eat anything in the laboratory and do not drink anything or do not smoke in the laboratory. So it is not at all allowed. Okay. So next is protect yourself from the shocks. So while you are working in the laboratory there are many apparatus like electricity. Okay, electricity, okay, electric wires. Okay, and so on. There are many machines. Okay, there are many machines are available. So what you have to do, you should be careful. You should not put your fingers in the electric board. You should not pull any wires. Okay, and you should not damage any machines. So that is very important. You have to protect yourself and also you have to protect the machines also. Okay. So next is keep it clean. Keeping the workplace clean after the work. Okay, and put everything back into the place and wash your hands. That is most important. Why you have to wash your hands after the experiment? For example, if you are doing some research or work, so there may be some kind of damage, some kind of bacteria may stick to your ear, to your hand. So if the bacteria stick to your hand, what will happen? When you are eating, that bacteria may enter into your body. Okay, so and it may cause some kind of diseases. So this is the reason you should not touch anything, and if you touch something or if you do some research or anything, so you have to wash your hands after the experiment. You have to wash your hands. Okay, that is the most important thing. So next is observation of cells. Okay, observation of cells is the most important thing. So what is observation? Observation of cells in the mastic is nothing but that we have discussed already. Okay, this we have discussed already in the previous topics. Okay, so mastic is also made up of cells. Okay, what is matchstick? Matchstick is nothing but plant cells. Okay, it is made up of plant cells. So you can observe matchstick with the help of of microscope. So our aim is to observe the cells in matchstick. Okay, cells in matchstick. Materials required are matchstick, water, scalpel, slide, cover slip, etc. So these all are the things that are necessary for this experiment. So procedure. What is the procedure? So first you have to soak a matchstick in the water. You have to soak a matchstick in the water for 30 minutes. So what happens if you soak a matchstick in the water? This is water. You have to put a matchstick for 30 minutes, and after 30 minutes you have to take it out. What will happen if you put matchstick in the water? If you put matchstick in the water, it will absorb water. Okay, it will absorb water and will become a little bit smooth, and it is very easy to cut. Okay, this is the reason we have to put the matchstick in the water, and we have to cut into thin slices. Means this is a thin slice of the matchstick. This is a matchstick. Let us say this is a matchstick, and if you cut on the top, you will get small thin slice. This is a thin slice, and you can use this thin slice of the matchstick to observe under the microscope. How do you observe? So take a slide. Okay, on the slide put this matchstick and add a drop of water, cover it with the cover slip and put it under the microscope and observe. Then you can see clear image about the matchstick cells. Remember, those cells are dead. Okay, those cells are dead, and those cells will look like empty spaces like this. Those cells will be in the form of empty spaces. Okay, so this will be about your observation. So take a slice with the help of a brush and keep it on the slide and put a drop of water, then cover it with a cover slip and observe under the microscope. And during this process, make sure there should be no bubbles on the slide. Bubbles should not be present. If the bubble is there, you cannot observe clearly. So observe under the microscope and draw the diagram, and also compare the diagram with the cork cells. So in the previous classes also we have discussed some cork cells, no? So those cork cells and matchstick cells, compare both. Okay, they look almost similar. Okay, so the living empty cells are observed under the microscope. Result is the cells in matchstick can be observed. Okay, so next one will be the observation of onion peel cells. How do you observe onion peel cells? So first is aim. Our aim is to observe cells of onion peel. Okay, so materials required are onion peel, water, slide, cover slip, microscope, and scalpel. So here what is meant by slide? Slide is nothing but a glass plate. What is cover slip? Cover slip is thin glass plate is known as cover slip. The slide looks in this way. Okay, this is slide and the cover slip looks like this. Okay, this is cover slip and this is slide. Okay, so here the procedure: take an onion bulb, cut into small pieces from it, and this is small piece. You can see this is small piece. Bend the small piece and peel it. If you peel it, you can see thin transparent membrane will be obtained. So this thin membrane is obtained, and cut the small portion of the membrane and put it on the slide. Okay, put it on the slide, add a drop of water and cover it with the cover slip just now as we have seen. So this is a slide. Okay, so put the small membrane and also cover slip, cover with the cover slip and observe under the microscope. If you observe under the microscope, you can see the cells. Okay, you can see the cells in this way. So this is the cells. This is the cells that you can observe under the microscope. Okay, and by observing under the microscope, you can draw the diagrams. So if you draw the diagrams may look in this way. Okay. So next one will be the observation of human cheek cells. So this is activity 3. So in this activity 3, here you have to see the aim. What is your aim? The aim is to observe human cheek cells. So materials are human cheek cells, slide, cover slip, spoon and also water. So the procedure is, first you have to wash your mouth. Okay, you have to wash your mouth, then take a spoon and scrape the inner side of the cheek smoothly to obtain the cheek cells. Means inner side of the cheek, there are very delicate cells. So with the help of spoon, just scrape it. If you scrape it, the spoon will have some kind of tissue. Okay, those tissue are known as cheek cells. So again, take a slide. Okay, put the cheek cells on the slide, cover it with the cover slip and observe under the microscope. Okay, if you observe, you can clearly see the transparent cells can be observed. So this transparent cells looks in this way. In this diagram you can see these are transparent cells of human cheek cells. So result is human cheek cells can be observed. Okay. So observation of nucleus in the cheek cells is activity 5. So in this activity you can see the aim. The aim is to observe the nucleus in cheek cells. Just now we have seen observe the nucleus, but now we are going to observe the nucleus in the cheek cells. So materials are cheek cells, cheek cells that you can take it from the mouth. Okay. Next stain is nothing but a color. That color may be saffron red or methylene blue. Okay. So these are the materials and the cover and also slide and also cover slip and microscope, water and also the filter paper. These are the materials that are required. Okay. So next is the procedure. How do you do the experiment? So wash your mouth, scrape a little bit of inner side of the cheek cells slightly to obtain the cheek cells. When you obtain the cheek cells, what you have to do? You have to place it on the slide and add methylene blue. So if you add methylene blue, so those cells are going to take color. Okay, those cells are going to take color and the color will be used for the staining of the nucleus, staining of the cytoplasm and other cell organelles. So after the adding color, so if there is excess amount of the color, just to add some amount of water to remove excess water and also after that observe under the microscope. Okay. So if you observe under the microscope, you can see the blue colored spots. So in this here you can see the cells. Okay, the cells are going to have some blue colored spots. Okay. So here this is a nucleus and this is a cytoplasm. Okay, cytoplasm and outer layer is cell membrane. Okay, you can see this diagram clearly in the textbook also. You can see this diagram very clearly. So result is the nucleus observed in the cheek cells. Okay. So next event will be in the plant cells the cell wall. What is meant by cell wall? Cell wall is nothing but the outer layer of plant cells. Okay, and also bacterial cells is known as cell wall. This cell wall is going to give shape, structure and support and rigidity to the cells. So cell membrane, in the cell membrane, the outer layer of the cell membrane where you can see. Cell membrane can be seen in the animal cells and also cell membrane can be seen in the animal cells only. But the plant cells will have the cell membrane. Around the cell membrane there is cell wall. So cell membrane and cell wall is present in the plant cells. Okay. In animal cells only cell membrane is present. Okay. So this is the difference. Okay, the cell membrane is permeable. Okay, semi-permeable or selectively permeable. It means it will allow only certain substances to pass through it. That is known as semi-permeable. So here the cell is going to have the nucleus. What is nucleus? The nucleus is nothing but inside the cell there is a structure. Okay, inside a cell, in the cytoplasm, there is a cell that is known as nucleus. So what is the nucleus is more important in the cellular functions. In the cells, it is going to do some important functions like processing of the information, commanding, and also help in the production of proteins. It is going to contain DNA and so on. This is the importance of the important function about the nucleus. Okay, so if you see the nucleus is periphery in the animal cells, in the plant cells, the nucleus is near to the adjacent. Let us consider this a plant cell. The plant cell is nucleus will be here, nucleus var here, and so on. But in case of animal cells, the nucleus will be in the center. Okay, in plant cells, the nucleus will be at the edges or at the periphery. Okay, so next one will be the cytoplasm. The cytoplasm is heterogeneous material. What is heterogeneous? Heterogeneous is nothing but it is going to contain the nucleus and also the containing cytoplasm and so on. All these things are present in the cytoplasm. Hence, we are calling it as a heterogeneous. So, cytoplasm contains different substances like water, cell organelles, gases, proteins, etc. These all are present in the cytoplasm. Okay. Next one will be the preparation of microscopic slide. How do you prepare the microscopic slide? So, first microscope is prepared. First point is microscope is slide is prepared on a 2 mm thick. Okay, 2 mm thick, and that is 3 centimeters into 8 centimeters rectangle strip, and it is clear and also clean glass piece called slide. So this is slide. So what is the size? It is 3 centimeters. So this is 3 centimeters and also 8 centimeters. Okay. So this is will be the about thickness, okay, and also this is dimensions, 2 millimeter thickness. So the size will be 2 millimeters in thickness, okay. So next is, if the object is thin and flat, it can be directly placed on the glass slide towards the center. So and also if you after placing the tissue on it, you can cover it with the cover slip, okay. And also in the center, if you add a drop of water, it will be more useful to observe under the microscope clearly, okay. So next, a drop of glycerin is added. Glycerin helps in the prevention of evaporation of water. Water will be prevented. Evaporation of water will be prevented with the help of glycerin. Okay. So the glycerin saves the material from drying out. It means dehydration will be prevented with the help of this material, with the help of glycerin. Okay. So if the object is thick, so cut it into thin pieces, and also nearly 0.5 mm or less than thick sections can be cut with the help of a sharp razor. Okay. If the object is transparent, it can be stained. What is transparent? Transparent means it is colorless. Okay. So if the object is colorless, you can use a stain. Stain is nothing but color. Okay. And also between the kinds of cells, you can observe all those things. Next is place the stained objects on a slide, and also a drop of water should be added and covered with the cover slip without formation of air bubbles. Okay. So air bubble should not be there. If the air bubble is there, you cannot see clearly. Okay. So remove excess of water on the slide using blotting or filter paper and so on. Okay. So this is the procedure. So slide, a drop of water is added through the dropper, then covered with the cover slip, and adjust with the help of needle and observe under the microscope. Okay. Next is the staining technique. What is staining? Staining is nothing but adding color, is known as staining technique. Okay, adding color to the tissue or any material. So based on the fact, there are few colored substances that get attached to different parts of the cell. So what is the parts of the cell? Parts of the cells is nothing but cell organelles. The cell organelles will have all the structures. They are going to take the color. Okay. So when they are colored, you can clearly observe under the microscope. Okay. And the cells in the highly particular areas of the cells. These coloring agents are known as stains. Stains are nothing but they are colors. So the process of adding color to the cells are known as staining. We can use this technique to observe several things like microorganisms, different parts of the cells, etc. So for this, we need to use materials like safranin, methylene blue, and also some kind of red ink can be used in the staining technique. Okay. So to make the safranin solution, how you can do? You can take 25% or 1/4 teaspoon of the safranin red in 100 ml of the water and dissolve it. It will become safranin solution. Okay. And I hope this lecture is helpful to you. If you have any doubts or questions, post it in the live chat section or comment section so that I can read and I can answer to your questions or doubts. And we will discuss the